Okay, welcome back. Now let's finish up on the chapter 1. And what we'd like to look at, of course, is naming, classifying, and identifying microorganisms. And I say we're going to greatly cut down on the amount of material in this section. First, I've already gone over, but I'll do it again. The bacteria and protozoa are single cells. Animals and plants are composed of cells, so there's one huge difference. Trillions of cells kind of working together. We have 100 trillion cells in our bodies and that sort of thing. Spherical, polygonal, cubical, cylindrical types of cells. So we have all sorts of different types together. They all contain a protoplasm with a cell membrane, have chromosomal DNA with a container around it, ribosomes for protein synthesis, and really complex functions. So eukaryotes, animal plants, fungi, and protozoa, contain organelles. These are the compartments that safeguard each of the containers from the protoplasm that's inside the cell where all the nutrients and various things are. Bacteria, they don't have what they call a true nucleus. Now, they have a chromosome and it's a double-stranded DNA that is circular, like a big rubber band that's all folded up. But they don't have organelles either. So think of it as a bathtub with all the components in it, like I mentioned before. It does have and contain a lot of the nanotechnology that we see in eukaryotes. So it's fine structure and very detailed, and they can engage a lot of the same activities, but they do it in a very stripped down fashion. They can accomplish it, they just don't need all the bells and whistles that the eukaryotic cells have. Now, the nomenclature. This is how I want you always to refer to when you see a bacterial name, and we have the genus and species name. The genus is the first name. So Escherichia coli is one I know well. It's been around for a long time, and Dr. Escherich is the one who identified it. And so Escherichia, we call it, is underlined and it's capitalized. And the coli, which is the species name, is always in lowercase. Now you can italicize it without underlining it, or you can underline it if you can italicize, but it has to be done that way. Now, you can write it as E period coli, and that still gets italicized or underlined. So that is the way we always refer to these microorganisms, and that's really where I want you to focus on the genus and species. I'm not really worried about all the domain, kingdom, phylum, division, class, order, family part of it, because my guess is a lot of this is going to change with the genome project, and it's going to be classified more from a DNA sequencing point of view. I don't think the genus species will change that much. It's my guess. I don't know. So here's the domain Eukarya, and you can see all eukaryotic organisms, and you can see it's pretty diverse, and you can see the kingdom Protista contains Paramecia, Histostomata, all these different types. So this is a Protista name, so you can kind of look at that. Phylogeny is the taxonomic scheme that represents a natural relatedness between groups of living things. So when you study the phylogeny, now, for an example, E. coli is closely related, they're close buds and cousins with Salmonella. So it's some Salmonella typhimurium and Escherichia coli evidently somewhere along the line had divergent evolution, and so they're related but not identical, and they have changed some characteristics. What that means is, from a diagnostic point of view, identifying it clinically, it provides some challenges, because now we have to distinguish between these very closely related type of bacteria, and they're responsible for different types of diseases, so we need to know. So we focus on identifying certain characteristics like sugar utilization or antibiotic resistance patterns or some of the other things to kind of focus on that to kind of inform us as to what they are, and that way we can bin it in the right bin and then try to treat it. So that's where we are on that. So we do see, as divergent evolution and through various structural and functional changes, and through natural selection. And of course, our antibiotics and our ways of trying to control these microorganisms have artificially kind of put a natural selection pressure on them, and so in doing so that we see evolution occurring kind of on a rapid basis. I call it microevolution, and I don't know if that's proper or not, but that's the way I kind of look at it. So, in the Universal Web of Life, Charles Darwin and Haeckel proposed two kingdoms, plants and animals. So Protista or protozoa, and Haeckel then added Monera, and in 1870, and that's where we're at with the bacteria. Whitaker model added fungi in the period when I know 59. Boy, that sounds like a long time ago. That's the year I was born. I'm a fossil, so you can just put me under the Whitaker model there as a fossil, based on structural similarities and differences. So there's different models. That's why I think in the long run DNA technology sequencing is going to change a lot of this, so we can look at the genetic relatedness based on conserved small unit ribosomal. So we use SSU RNA as a way for the Woese-Fox system of taxonomy, and we look at the sequences that reveal separate groups of the bacteria called Archaea, which we know is the older, and we have the new system, bacteria, Archaea, and Eukarya, based on looking at the ribosomal RNA. And so, as I mentioned, so genetically is going to become more and more and more. So everything after this talks about, for those who have different needs and things, you get the longer descriptions of the graphics that we present in the video, so you'll always have that. If you need it, it's there. So that's pretty much it, and the takeaway is, let's write the organism's name in italics or underline, capitalize the species and underline or italicize, and lowercase for the genera, and we're in good shape. And then the rest of it, well, I'm not too worried about it. We'll go from there. And the next chapter we're going to discuss will be chapter 11, and we'll get on to that.